Okay guys, here is our next fun sheet. It is 3.1, hypothesize that the impact of structural changes to genes located on chromosomes may result in harmful, beneficial, or neutral effects to the structure and function of the organism. So that's a whole lot of something. But before we can talk about all that something, we got to talk about what genes and chromosomes are. And we have started talking about them since we've been talking about cells since the beginning of time. So we're going to revisit some information we've talked about already, and then we're going to learn some new information. And my cat is all about this tonight, so you've already heard her meow, so you might hear some more. She likes to headbutt me while I'm writing also, so excuse the gibberish. And I tried a new pen on the iPad today, and that's a little questionable too. Alright, so our first term is DNA, which is the abbreviation for deoxyribonucleic acid, and it's what chromosomes are made of. They contain genetic material that determines our inherited traits. DNA is also sometimes called the double helix, and also sometimes the more generic, not scientific term, twisted ladder. And we're going to draw those and talk about those more in depth in a little bit. Now our next term is chromosomes, and we have definitely been talking about chromosomes since the beginning of time. Oh wait, let me go back to found where. Where is DNA found? Do we know? Well, we're going to find that out in a second. So hang tight. So chromosomes are made from DNA, and it's where genes are found. They carry our genetic information. You know, or you should know, that DNA makes up the chromosomes, and that our chromosomes live where? Do you guys remember? What cell organelle that we've been talking about since the beginning of time? Oopsie, that wasn't supposed to happen. Let me try again. Yeah, the nucleus. It's found in the nucleus. It's the control center. That's why it's the control center. That's where all the genetic information is that controls everything about us is. It's in the nucleus. All right, so that's chromosomes. And then the chromosomes have genes on them, and the DNA codes for these genes. And genes, back up. I forgot about the tightly packed DNA. So DNA is super long. If we unraveled it, it would be super long. And I could tell you how many feet it is, but I forget. So I'm not going to make it up. But it's long. And it gets tightly compacted. And when it ravels up and shrivels up, it makes the chromosome. So if you ever had a hose on a wheel and you unwound it all the way out, that'd be like unwinding the DNA. And then when you wheel the hose back up, that's tightly compacting it. Well, that's what's happening to make the chromosome. Okay, on to our genes. So genes are instructions for an inherited trait, and those live on the chromosomes. Okay, I'll show you an illustration here in a minute. I think I forgot a term, so you're going to have to squeeze this word in somewhere. Oh, hold up. So genes live on sections of DNA at a specific location of the chromosome that determine our inherited traits. That's basically just what I said above in different words. Write it down also. Okay, now here's the word I forgot. I forgot inherited traits, so I want you to write this somewhere. Inherited traits are characteristics passed from our parents to the offspring. So your parents gave you blonde hair or brown eyes or freckles or dimples. They're the characteristics that you got from your parents. And that's all coded from the DNA that lives on those genes. Okay? So make sure you added that term. All right, nucleotides. So nucleotides make up DNA. A nucleotide contains a sugar, a phosphate, and a nitrogen base. Nitrogen bases are the steps of the ladder if we're talking about the DNA and we're considering it a twisted ladder. The steps are made up of the nitrogen bases. And so the part I didn't tell you was that the sugar and phosphate make up the arms of the ladder, or they sometimes call it the backbone. Okay, so the sugar and phosphate make up the arms of the ladder, also known as the backbone, and then the nitrogen bases make up the steps. So I'm going to draw those nitrogen bases in there. And those nitrogen bases are adenine, thymine, guanine, and cytosine. And those, I'm going to teach you about how they're always together in a second. But those are the only 4 that are in the DNA molecule. And they have to pair up a certain way, okay? So make sure you drew that little picture and you labeled sugar phosphate as the arms, and then the ATGC as the bases. Okay. So I went back and added it. So next to sugar, right arm, and next to phosphate, right arm. All right, so next we have complementary pair. And this is a whole lot of words and have to draw a little bit. So you might want to listen first and then rewind it and watch and write again, because I kind of have to jump around a little bit to explain it. But we're talking about the nucleotides, okay? A nucleotide is made up of a sugar, a phosphate, and a nitrogen base. And so adenine always pairs with thymine, and guanine always pairs with cytosine. That's a complementary pair. A and T, G and C. A and T, G and C. The way to remember what goes together is at Greenville City. Okay, you go to school at Greenville City. AT, always together, makes the word at. Greenville City, GC. All right, so down below I have written this strand of letters. So think about the DNA strand, the double helix. Miss Foshi and I will show you a model of one, and it's a twisted ladder or colorful steps. And so each of those letters makes up part of the steps. And so A, T, T, G, C, A, C, C, G, A, T, it's just a random combination of letters. I've made that up. It doesn't really mean anything. But its complementary pair is going to be what letter matches up across from it. And I already told you that A always pairs with T. So looking at this, A pairs with T. So the same thing happens for T. T has to pair with what then? A. So you have another T. What is it going to pair with? A. G pairs with C. So what's G going to pair with? C. And then C pairs with G, and A pairs with T, and C pairs with G, and C pairs with G. Figure out the rest. What goes with G? What goes with A? What goes with T? Were you right? Okay, so that's a complementary pair, the letter that goes across from it. All right, so the next thing you need to know about this, and you need to be writing all this down, is that a group of 3 of these bases make an amino acid, okay? And then when you have 3 and then 3 and then 3, and if I was paying attention, I would have counted and added it so it was an even set of 3s, okay? When you have a string of amino acids, they're going to make proteins, okay? And so proteins code and give directions for a lot of stuff. Okay, that's why when we think back to cells and cell organelles, ribosomes make proteins, and that's why there's so many ribosomes in cells, because they're making proteins for all kinds of different stuff. Proteins are the building blocks for a lot of things, including our DNA. And if you remember, the ER has the rough ER, and it's those ribosomes are on there. Well, that's because it's modifying proteins, and proteins, the ER also deals with DNA stuff, and a whole lot of complicated stuff I'm not going to get into right now, or this video would be way too long. But that's why they're there, because that's what's going on in the ER. All right, so make sure you wrote all this down. Rewind it and make sure you get this written. All right, so next we're going to talk about the stuff that basically our SPI was talking about in the very beginning. So mutations. So think about the DNA molecule, the A and the T and the G and the C on that twisted ladder, as a big zipper. And if I could pull it apart, then you would have half of that complementary pair on the one side. Those letters we just wrote would be on the one side, and then the other half would be on the other side. And what happens is they go through a process of replication. In cell division and mitosis, we have to make copies of our DNA. And so when that happens, it makes an exact replica. And then the one side matches up with the other side and forms a new one. And then the other two that are left over form a pair. And so that's how you get DNA in the new cells. Well, sometimes the zipper misaligns. Think about it. Sometimes when you're zipping something up and it gets off and the teeth don't line up right and you have like a weird bump in the zipper, or some clothes get stuck in it and it messes it all up. Well, it's the same idea-ish that happens with what could cause a mutation. So mutations is a change in the number, the type, or the base order on a piece of DNA. So when that happens, different things can occur. And we're going to talk about these later on in class in more specifics. Like if something's deleted, it's going to cause this. And if something is substituted, it might cause this. But this is the broad topic, just to make you familiar with these words. The results of mutations could be beneficial, meaning it improves the organism or helps it. Neutral, meaning there's no change, it doesn't matter that you have this mutation or not. Or harmful, meaning it causes damage or death or sickness. So one of the terms is deletion. So it would be you're taking one of those base letters, an A, a T, a G, and a C, and you're taking it away somehow or another. It doesn't meet its match, and so it's been deleted. Insertion, a base has been added, so there's an extra base in the chain, so it's messing up the chain. Or a substitution, the wrong base is used. So, I think I put a picture in here. Let me see. Okay, so here, if you look at this zipper, you can see, let's just say that those yellow ones are the A's, and the blue ones are the T's, and the red is the G, and the green is the C. So, red, green, blue, yellow. Okay, and so you can see how this first one all matches up. The yellow matches the blue, the red matches green, blue, yellow, blue, yellow, blue, yellow, red, green, blue, yellow, blah, 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 blah. So that all matches up. But then if you look down here at this one, can you see where something's not matched correctly? So right here, this red should be with a green, but instead it's with a blue. That is a mess up. Okay, it substituted the wrong base in there, and that's going to cause a mutation. It's going to cause something, and we're going to talk about that in class. Things that cause these mutations can be physical or chemical agent that causes the mutation. It can be because of genetics, or it can be environmental. Sometimes UV radiation causes changes in our DNA. Sometimes chemicals you're exposed to would cause changes in our DNA. Sometimes it's viruses that would cause a change. So they're called a mutagen. All right, so on this bottom part of your paper, find some room to draw. You're going to draw these examples. So first, we're going to draw this chromosome, okay? The chromosome is the X part. The X is the tightly compacted DNA, okay? So you have this big DNA strand, and it coils up, and it makes the X, which makes the chromosome. And then these little red and blue bands on the chromosome are our genes. So genes are what code for our inherited traits. Example of an inherited trait would be hair color, eye color, freckles, dimples, hitchhiker's thumb. The list could go on and on and on. So the X is the chromosomes. That's what I just went back and highlighted there. And then now we look down here, we have this double helix, the DNA strand, okay? So draw a double helix and label it DNA. And then draw something that looks similar to this strand of DNA that's showing you how it's unzippered, and label the arms of the ladder as sugar and phosphate. And then label the rungs of the ladder as the nitrogen base. I think we need to write, maybe not with yellow. Yellow is hard to see. The rungs of the ladder are the nitrogen bases. So draw that example there. And then the pictures on the right are just breaking it down for you more, showing you what the arms really look like, the sugar and the phosphate. The phosphate is this long purple thing, and the sugar is the pentagon shape, I think, 5 sides. So 1, 2, 3, 4, 5 sides. So this pentagon shape in purple is the sugars, and then it connects to the bases right here. So draw these pictures as best as you can in the area and label the things we did. You definitely need to draw a chromosome. You definitely need to put genes on your chromosome and label your genes. I mean, your pictures don't have to be Picasso's. Just get the parts on there and label the information. There's your gene. Here's a chromosome. Okay. So get that on there. And then do draw a double helix and do draw and label this part here. All right. That's it.